rhizome, the underground rhizome may be broken down into small pieces by chance. Then each piece can develop into a new plant body of polytrichum. Next is the sexual reproduction. So here you are seeing the vegetative reproduction. It is by fragmentation, the lower third one you are seeing. Vegetative reproduction, it may take place by fragmentation with the help of rhizomes. Sometimes it may take place with the help of protonema also. Protonema are the structures which are developed when spores will germinate, not on the highly developed plant, but after spore germination, the structure which is formed, that is called as protonema. In other bryophytes also we discuss this. So on the protonema, the filament-like structure is formed. On that, sometimes buds will grow, small buds at the in between the cells. Each bud, it develops into a new plant body. So that is the apical one. It can develop into a independent plant body of polytrichum. That is a gametophyte plant body. So these are the protonema and the fragmentation method, vegetative method of reproduction. Next is sexual reproduction, which is very, very important. Plant body is, polytrichum plant body is dioecious. Dioecious means that is unisexual. Male plant and female plant are different. Male plant will produce antheridia and female plant will produce archegonia. So, where they are produced at the tip of the stem, like structure, antheridia are developed. So, they are developed in groups. That's why it is called as antheridial head. The antheridial head are born in the axillary clusters of the tip of the leafy stem. So from the sides also antheridia will develop, so branches. So leafy stem they develop. They are surrounded by rosettes of leaves. The closely arranged leaves are present, and these rosettes of leaves are called as perigonial leaves. So this point you have to remember. Perigonial leaves are present surrounding the antheridial head. These leaves are different from ordinary vegetative leaves. Just they'll give protection. The perigonial leaves are spirally arranged. The antheridia are produced in groups in the axils of these leaves. So many antheridia produce. I'll show it that in the diagram. Thus the antheridial head will have different antheridial groups. In between the antheridia, to protect each antheridia for that is they may be crowded, so that's why to protect each antheridia, in between the antheridia sterile structures called paraphysis will develop. As you know, each antheridia is a club shaped structure. It consists of a small stalk. So small short stalk is present, above which club shaped antheridia produced. Each antheridia body of antheridia is surrounded by a jacket wall. Inside the jacket wall, many cells are present. Each cell is known as androcyte mother cell. This androcyte mother cell, it metamorphoses into a biflagellate sperm or the antherozoite. So here in the diagram, you're seeing the antheridial head. Left hand side diagram, first you concentrate. It is the antheridial head. This antheridial head, you are able to see the many antheridia and surroundings cells, the outermost cells, that is called as perigonial leaves. And in between the antheridia, sterile structures called paraphysis are present. So one of the antheridia, mature antheridia is shown in the diagram, through which antherozoids which are coming out, that is shown here. So That is about the antheridium development. That is antheridia, how they are formed. Then antheridia development, you're seeing in the second diagram, you're seeing the male plant body, and beside that, you're seeing the development of antheridia. First one is the antheridial initial cell that will divide longitudinally, and that cell and produces another mitotic division. First mitotic division, second mitotic division undergoes. Due to that, it produces, that is actually a periclinal division will takes place, and that will give rise to the wall of antheridium, and inside the cells which are formed there, that will be the androgonial mother cells. These androgonial mother cells, they divide and redivide and produces the androcytes. Each androcyte develops into a, that is antherozoite. In the lower diagram, the last diagram, what you're seeing is the biflagellate antherozoites. So that is the lower black color diagram. What you're seeing is that antheridia with paraphysis, and black colored, that is perigonial leaves. So this is all about the antheridia development head. Then about the archegonia. Archegonia developed at the tip of the stem on the female plant body. Male and female plants are different. So each that is archegonial head. Lower diagram, you concentrate. There you're seeing petal-like structures, black. What you're seeing is the leaves. So these leaves are called as perichetal leaves. Here is not mentioned, but that is perichetal leaves. Inside the perichetal leaves, a whole of leaves are present. On at the tip of the archegonial head, you will see flask-shaped structures, which are nothing but the archegonia. Many archegonia are formed. Here also, for the in between the archegonia, sterile structures, paraphysis are present. So each archegonia is a flask-shaped structure with a basal swollen part, that is called as venter, and the upper part long neck you can see. So, and the tip of the neck, you're seeing the neck cannot, that is, sorry, the tip of the neck, you will see the lid cells or cover cells. Here in each archegonium, in the venter region, at the lower side, you will see the egg cell, and the upper cell is the ventacanal cell or VCC, it is called. In the neck is very long here. So in the neck region, so many neck canal cells you can see. Neck canal cells, they are also called as that is NCC. That is 6 to 8 neck canal cells may be produced here, at the tip of which lid cells or cover cells are present. About the development of archegonium. Archegonium develops from the right hand side toward the left. That is arrow mark is there, that you concentrate. First, from the one of the initial cell on the archegonial head, so many initial cells may be there. That is called as archegonial initial cell. This archegonial initial cell, it divides periclinally, and it produces a central cell and peripheral cells. These peripheral cells will produce the neck and venter wall, and central cell, it develops a lower cell of that, it divides again, central cell or the axial cell, again it divides and produces the neck initial, and the lower one is called as venter initial. Venter initial, again it undergoes mitotic division to produce the egg cell toward the lower side, and ventacanal cell toward the upper side. Similarly, neck initial, it divides and redivides and produces 6 to 8 neck canal cells. So this is about the development of archegonium. Then about the fertilization. As you know, after the complete maturity of the archegonium, during favorable conditions, when plenty of water is available, that time antherozoids. First, antherozoids will mature and they'll produce the antheridium will mature. Antherozoids are released by the breaking of the wall of the antheridium jacket wall, and these antherozoids they biflagellate. They are released in group, they'll swim in water, and somehow they'll reach the archegonia due to the chemotactic moment, because this archegonia, mature archegonia, it produces a mucilaginous substance by the disintegration of the neck canal cells and ventral cell, and it produces pressure on the lid cells or opercular cells. Due to that, cells they are loosened, and they, along with water, antherozoids will enter. One of the antherozoid it reaches the archegonia venter region, and then fertilization will takes place. Antherozoid along fuses with the egg cell to form zygote, that is a oospore. Zygote is also known as oospore. So this oospore or zygote, it develops into the sporophyte. So about the sporophytic plant body. Here the sporophyte is developed on the gametophyte. So sporophyte is dependent on gametophyte for its survival in bryophytes.